you. Thank you for inviting me. It's been a really interesting morning hearing about the different standards that are being implemented. And it's really exciting when we think about the work that we're doing at Cohen Veterans Biosciences. And so I think this will actually fit in really well to be able to take what has already been discussed and focus it on one of the problems that we're focused on, which is looking at biomarkers for post-traumatic stress disorder and traumatic brain injury. So let me get a laser pointer here. All right, so let me just do a very brief introduction of Cohen Veterans Bioscience. We are a nonprofit biomedical research organization and technology organization. And really our relationship is with both academic, government, and industry is as an integrator. We look to work with all of the different stakeholders in the PTSD and TBI fields as a way to bridge the discoveries and early work that's being done in these areas. We like to add evidence, de-risk, as we've been talking about some of the efforts that have been done, and so they can move over to the industry side and be taken up in terms of development of biomarkers for drug development, diagnostics, and therapeutics as well. And so that's really our main goal. We do look to employ a mechanism-based framework as a basis for neuropsychiatric disorders. And this is not what is done at this time, as I'm sure everyone knows. We are now we have questionnaires that are given to us by clinicians, and we are asked questions, subjective questions, instead of having a biologically based way to assess if someone has some type of psychiatric disorder. And so we want to move over to a more mechanism-based approach to characterizing these symptoms. So, oh, sorry. So why are we focusing on these areas? This is a huge unmet need. When we look at what has been done, at least in the US, and it's pretty standard across the rest of the world. In terms of approved diagnostics, there are none for PTSD at this time, and there are some for traumatic brain injury. However, these are aids in diagnosis, and so they're not a standalone way to diagnose traumatic brain injury. Usually they just will tell you if you do need to get a CT scan. In terms of therapeutics, there are 0 therapeutics for traumatic brain injury. And in terms of PTSD, we have 3. 2 of those are SSRIs that were approved about 20 years ago, and they have limited efficacy in individuals that have been diagnosed with PTSD. One of the newest therapeutics is actually a modification to your Apple Watch. What it does is it has biosensors that will detect if you're about to have a nightmare, and it will wake you up before the nightmare happens. So it doesn't actually treat the disorder, it will just stop the symptoms from occurring. So as you can see, there's a highly unmet need and a real need for us to provide more evidence so industry will follow and develop therapeutics in these areas. So in terms of the patients that we're focusing on, now I want to really focus here on PTSD as we move forward in the presentation. We know that a large number of people do experience a traumatic event. Many experience multiple traumatic events. Of that, when we take a large look at it, around 8% of people who experience a traumatic event will develop PTSD. And then there are a large number of individuals who may not meet that diagnostic criteria, but still have some symptoms. Now, these are the different symptom clusters. These are what the physicians will ask about when you may be telling them about your symptoms: is intrusion, avoidance, cognition and mood changes, arousal and reactivity changes. So as you can see, there's many different types of symptoms that you can get with this disorder. So what are the challenges and needs? What is industry challenged by and what is it looking for? And we're working with our partners to help define these and then hopefully bridge these gaps. One is that, as you saw, there was a lot of symptoms for this disorder, and you get over 600,000 symptom combinations. So this means that it looks different in almost everyone. You have different combinations in different individuals. It depends a lot on the person. So you have significant heterogeneity. And this is a challenge when you're trying to develop therapeutics when the symptoms look different in each of your participants. Um, a second huge gap is the lack of diversity in studies. So for example, at Cohen Veterans Bioscience, we have sponsored a number of cohorts and continue to, in which the participants are largely veteran populations. And so this is mainly males, so about 90 to 100% male participants. Um, and they have a very specific age group. And as you may imagine, the trauma is not selective. And so there are many other individuals that we need to understand more about how trauma affects them. If we think about children, older people, women, all of these populations need more study. So there's large gaps in our understanding of how trauma and subsequent PTSD is affecting these populations. Um, so when the pharma partners look at this and they have drugs in their therapeutic pipelines, one of the things they'd really like to do is stratify patients. They want to understand, are there certain groups that have specific symptom clusters that we may be able to target with our therapeutic? There may be people that respond better than others. And this has been seen with some of the currently developing therapeutics that we are able to we are able to tell maybe who will respond. And so that's very important for their clinical trials. For all neurological disorders, target engagement assessment is always a challenge. And the ability to objectively measure the efficacy of the treatment is a high need, and it'd be something that they would really value. So from this, as you can tell, getting biospecimens and biomarkers are really required to address these issues to help, one, give us that biologically based understanding, but also to allow us to de-risk these potential programs in our industry partners pipelines so they're able to develop these. So for post-traumatic stress disorder, the collection of cerebral spinal fluid or brain tissue is not an option. And so we look to peripheral specimens. And so I'm going to focus on blood samples. Now, there are some other matrices that we're looking at as well, things like saliva, but we'll focus on blood for today. When it comes to those kind of specimens, there's so many unknowns. And so, thinking about the participants and what might be going on in them is one factor that we need to consider. In addition to all of the other aspects that we've already discussed today. And so I'm going to go into this just in a few slides. So in addition to the unknowns that we have from blood samples, we also know that measuring biomarkers from neurological disorders is a challenge in this type of matrices. For one, in the psychiatric disorders, the biomarkers we may be looking to identify may not be a pathological sign. So there may not be a mutation, there may not be an aggregated protein. It may be something, for example, like cortisol, where it's naturally in your body, but it may just be at a different level. So you're looking for something that is already naturally there. Another aspect is that brain-specific proteins in the blood are at very low levels. And so unless you have some type of injury or a neurodegenerative disease, they're going to be very low. And so you need, very good assays in order to measure these proteins or metabolites, whatever type of biomarker you're looking at. And then we have to account for the rest of the body that is, adding in to the circulation, if you have a liver condition, a heart condition, any of these aspects can cause fluctuations in potential biomarkers. So you need to be able to account for that. So let's dive in to these potential biomarkers that have been reported thus far. I like this figure because it shows us a number of different biomarkers that have been shown that they seem to make sense mechanistically if we think about things like stress, things about hyperarousal. For example, one piece is looking at immune markers. There's a number of reports showing inflammation. So the issue is that a lot of these, there's a lot of discrepancy. And so it's very unclear, what type of dysregulation there is. And so this may lead back to some of the things we talked about earlier in terms of quality samples, quality collections, that these unknown factors may have led to some confusion. And so let me make this a little clearer. So this is from our PTSD biomarker database at Fraunhofer. And what it is it's, a number of the most highly reported biomarkers for this disorder in plasma. And that's across the x-axis. On the y-axis, you see the total number of reported studies. And so let's look just at BDNF here on the left-hand side. I believe it's for you. But so it's brain-derived neurotrophic factor. Um, this one we see in 4 of the studies, it was found to be increased in individuals with PTSD versus control. In 5 studies, it was found to be decreased. And then in 8 studies, there was no change at all. So when you see these types of reports, it's very difficult to know what exactly is going on and how would you target this with a therapeutic. And there's lots of reasons for this. These aren't necessarily wrong. One may be different assay methods. So some of these, maybe they're not sensitive enough to detect the changes that you would expect to see, or maybe not specific enough to detect the protein or biomarker of interest. I think one of the largest things is the variable clinical study conditions. As I mentioned earlier, we have our veteran specific cohorts, and there are a number of other cohorts. And so, for example, if we had, say, children who had a traumatic experience or people from an emergency room who had, say, some kind of accident or some kind of assault, they're going to look very different from, say, a veteran population that we have in our cohorts. So comparing them doesn't really make sense. So we need to have a really good idea of who these participants are so we can make sense of what these results might mean. And then last thing is a huge factor, the confounding issues. So this could be things like medication, it could be co-occurring injuries. So for example, if I were to look at something like, one of the inflammatory cytokines, if we looked at someone who had been in the emergency room and they perhaps had an accident and broke their leg as well, is it due to some other system injury versus is this from PTSD? So we need to really have a good record of these types of information. So let's look a little bit more at this brain-derived neurotrophic factor as kind of a case study for what I'm trying to convey. So this protein, it is important for synaptic plasticity, and it's essential in learning and memory. It is involved in the pathogenesis of PTSD, and there are a number of industry groups that are developing NMDA receptor antagonists that are they're primed to increase BDNF levels. And so this will increase plasticity, and hopefully with therapy, you'll be able to have things like fear extinction and help process through the PTSD. So for example, ketamine, which is something that has been in the news quite a bit lately in terms of some approved therapeutics. So it is a NMDA receptor antagonist. And what it does is, by inhibiting the NMDA receptor, you get through downstream signaling an increase of BDNF, which increases synaptogenesis, plasticity, and hopefully improves your symptoms. So there are some issues though. So one, if you are developing something like this in your pipeline, it is something that you would want to measure. Are we able to effectively increase our BDNF levels? Are we seeing changes, of these? How long do they last for the participants in the study? So these are very important questions. Now, we do know, though, that collection details are really, really important. So if we look at serum versus plasma, we find that the concentrations are very, very different. And so you can't, if depending on the tube you use, you could have very different levels of BDNF. So these pre-analytical factors are key. Another thing we found is that we did not find this, but it's been reported that the circadian variations are significant in terms of BDNF. If you see this graph on the right, on the top right, we've got the different time points, and these are males, and so they had their blood taken at these different times of day, and we have the BDNF concentration on the y-axis. And as you can see, the BDNF actually, it's highest in the morning and then goes down throughout the day. So depending on what time you took the sample, it may actually affect how you interpret your results with the therapeutic that you're looking at. Another example of this that is a fluctuation is the hormonal status. On this bottom graph, we can see this, it's an ovulation cycle graph of a number of ladies. So we have the BDNF concentration on the y-axis, and then the day of the ovulation cycle on the x-axis. And you can see that it fluctuates greatly throughout that process. So these types of factors are so important when you're trying to interpret any results that you have specific to BDNF here. So how do we proceed? I think, the guidelines that have been set up, the best practices that have been recommended, if these can be followed, this will help us really find some meaningful answers. If people are able to follow standardized collection and storage procedures, we've found that this can be really, really helpful, even for things that are very tricky. Some of these biomarkers are a little bit less stable than others. However, if you do use a standardized protocol, we are able to compare across sites, across different cohorts. And as I mentioned, we lack diversity. We need to have an understanding of a larger swath of the population. And so it's really important that we're able to compare from different sites. And then be sure to have the relevant participant information disclosure that is included on the consent form. Like I said, it's difficult to make interpretations if we don't know some things like, say, medication or other co-occurring conditions. So if we have the ideal conditions, these provide the confidence and the ability to understand our results better, and then provide this information to our industry partners, definitely with more confidence. So our top line takeaways for today is that one, partnering with diverse biobanks is absolutely essential for this research, and we really need to understand these different populations, these different types of trauma, to find effective therapeutics for people. So the more information we have, the better. Now, I know not everything works out perfectly, and I know that we have limitations, we have trouble with bandwidth. As long as it's detailed, say, okay, we couldn't get the sample to the biobank, for an extended amount of time, as long as, that's flagged in the notes and we're able to understand, maybe what the limitations of the biospecimens are, we can take that into account. If we have more information, it opens it up for more uses for that potential biospecimen to develop biomarkers for the drug development process. Consistency and attention to detail are essential for us to really understand what's going on in this disorder and achieve success. Uh, Thank you for your time, and I look forward to answering any questions.